Hey guys, today I'm going to talk about Angelman syndrome. This is Dr. Harry Angelman, an English physician, and in 1965 he observed 3 children that were unrelated but showed similar symptoms of a certain disease. These symptoms included severe intellectual disability, motor disorders, lack of speech, and seizure activity. This painting is called Boy with a Puppet, and it was painted in the Renaissance in around 1520 by Giovanni Francesco Corrado. This painting inspired Dr. Harry Angelman to put together what he saw, and it inspired him to write a paper in which he referred to these children as puppet children. Now, this name was a little bit derogatory, so this name was later changed to Angelman syndrome. And then in the 1980s, this is really where you see Angelman syndrome pop up in the scientific and medical literature. So a quick explanation, what is Angelman syndrome? So Angelman syndrome is a genetic disorder that primarily affects the nervous system, and it's caused by a mutation or an abnormal function of the UBE3A gene in chromosome 15. Most of the time, this is not an inheritable disease. It's not inherited, but when it is inherited, it's inherited by the mother with paternal genomic imprinting taking place. So the gene that is inherited from the father gets inactivated. So symptoms and phenotypic effects. And one of the problems or one of the difficulties of diagnosing this disease is that many of these symptoms very much overlap with other diseases, including autism, cerebral palsy, Prader-Willi syndrome, Pitt-Hopkins syndrome, etc. So misdiagnosis often occurs. But to list them out, you have severe intellectual disability, delays in development, and delays in development usually start happening or start becoming apparent at ages of 6 to 12 months old. You have severe speech problems, so many times patients cannot speak at all, or they can speak to a very minimal extent. Problems with movements and balance, ataxia happen. There's inappropriate laughter, children being excessively happy, and hand flapping. So these patients are have a very happy demeanor. You can also see this in the pictures below. Very happy, excessively happy, and this is something that kind of gets a little bit more controlled and lessens over time as patients get older, but it is a common characteristic of patients with Angelman syndrome. You have seizures, EEG abnormalities, so the electroencephalogram, which shows very high spikes and amplitudes in the frontal lobe of the brain, sleep problems, tongue thrusting, microcephaly, which means having a very small forehead, strabismus, which means that the eyes are not properly aligned, feeding problems, flexed arms or arms flexed at the elbow, kind of at a 90-degree angle, wide-spaced teeth, a wide mouth, drooling, a protruding tongue, and a fascination with water. If I can change this slide. All right, so here's another picture that kind of shows some of the facial features of Angelman syndrome, including some features that I haven't mentioned, such as a sunken nasal bridge, a small chin, and an ear-to-ear smile. And here you can see kind of what the facial characteristic would look like. So DNA and protein changes that lead to the effect. So as I said, Angelman syndrome is caused by an improper functioning of the UBE3A gene on chromosome 15. And most of the cases are caused by specific mutations of the gene or of a section of chromosome 15 in which the gene is found. But there's also some cases where the cause is unknown. So we don't know what caused it. We don't know what happened to lead the patient to have Angelman syndrome. So why is a problem with UBE3A such a big deal? Well, UBE3A labels or tags other proteins that need to be recycled in the cell. And it does this by adding ubiquitin to these proteins. It ubiquitinates the proteins. This is, of course, very important because certain proteins need to be recycled, degraded, and also the cell is constantly turning off or on proteins to make sure that these cellular processes are happening well. So if UBE3A is not working properly, you have abnormal tissue development and abnormal development in general. So we have a paternal copy and a maternal copy of the gene. That should be fairly obvious that we have a copy of UBE3A from our mom and from our dad. But what's really interesting with Angelman syndrome is that in certain areas in the brain, certain tissues in the brain, there is genomic imprinting. So only the maternal copy is being activated and the paternal copy is inactivated. Now, of course, this leads to the maternal copy to be extremely vulnerable because if anything happens to the maternal copy, then that has a direct effect to the person. So when there are mutations happening in the maternal copy, that leads people to have Angelman syndrome, and that's what the disease is. What can these maternal mutations be? Well, there's a couple of them. So first you have the deletion mutations, and this is pretty simple. The UBE3A gene is deleted and it's not present. You can see this here, this blue area that would normally be there. It's just not there, and this constitutes 70% of all cases of Angelman syndrome. Then you also have just your general mutations, the general changes in the UBE3A gene, and this can be the expression of the UBE3A gene is what's most affected here. So as you can see in the picture, the gene is there, but there may be mutations that lead to the wrong type of protein being expressed. The protein may not be expressed at all, or it may be abnormally expressed, and that constitutes about 11% of Angelman syndrome cases. Then there's also the imprinting center defect. So every chromosome has an imprinting center, and this basically regulates what genes are going to be turned off and what genes are going to be turned on. So in 6% of Angelman syndrome cases, the problem here is that the gene, the UBE3A gene, may be perfectly well functioning and it's there, but the imprinting center turns it off. So what happens is you have the paternal copy that's being turned off because of the imprinting, but now you also have the maternal copy being turned off. And this, of course, leads to Angelman syndrome. Then there's also the paternal uniparental disomy, and this is a little bit more complicated. So here you have two copies that are being inherited of this gene, UBE3A, from the dad. And what happens is both copies are still going to be silenced. And this, of course, leads to Angelman syndrome because now you have no UBE3A gene that's activated and can do its job. This constitutes about 3% of Angelman syndrome cases. So the inheritance pattern of Angelman syndrome, and this is where it gets a little bit complicated because it is a complicated disease. It's not always inherited, and when it is inherited, there's a lot of other factors playing a role. So this is kind of a simplification and one of the ways that it could become inherited. So again, it's mostly de novo or sporadic, which means that they're just random changes or mutations happening in embryonic development or in the formation of gametes, the sperm or egg cells. So no inheritance is happening here. But in the cases where there is inheritance, there's imprinting going on. So if a male is a carrier, as you can see here, he may have that gene from his father, which is silenced. And when he passes that on to his children, he will produce no affected offspring because his gene will be silenced. But as you can see here, some of the offspring, this female and this male, will be carrying the gene. And what happens is when you have an affected female or a carrier female, and she passes on the mutated gene, she will produce affected offspring because her gene will be activated. So you can kind of see that here, that a carrier female has a risk of 50% of passing on the disease, because she still has, if she's a carrier, she still has a normal, healthy gene. So this is, again, sort of a simplification because there's a lot of other stuff going on. It's a complicated disease. So the treatments, unfortunately, there's no genetic therapy or cure available for this disease, but what we can do is we can help these patients with the symptoms and help them become more integrated into social life. So one of the things that can be used to help the symptoms are anticonvulsant drugs. If I can move myself here, the drugs here that you can see, those are anti-seizure drugs. So anticonvulsant drugs that help prevent seizures from happening. Many of these patients have sleep problems, so adhering to a strict sleep schedule can help going to bed better and sleeping better. You know, going to bed at a regular time, waking up at a regular time, that helps with regulating sleep schedules. And then there's also motility drugs, because many of these patients have digestive issues, which means that these drugs will help the downward movement of the food. Then there's also surgery for some of the problems and some of these symptoms, especially the facial or the physical characteristics, such as surgery for strabismus, which is the improper alignment of the eyes, that can be helped with surgery. Then there's the ankle braces, supports, and physical therapy for the mobility and walking, as you can see here in this picture, ankle braces. And then there's behavioral therapy, because these children are intellectually impaired, and the sooner you can help them out, and become a little bit more integrated into social life, a little bit more social, and help them develop their social skills to the extent that they can, the better. So you can see in this picture that this guy is helping this patient, this little boy with Angelman syndrome. Um, he is kind of playing with him with toys so that he can develop his motor skills, and also that he can develop a little bit his social skills and his communication, his nonverbal communication with this guy. And of course, because these patients cannot speak, and most of the time, cannot speak at all or speak to a very limited extent, you can help them with technologies and strategies to communicate non-verbally. So this is an example of that. This is a computer, an iPad that works with pictures, and that helps these children or patients communicate. And there's also other technologies that are coming out, such as voice emitting devices that help these children communicate, even if it's non-verbally. So the people affected. So this disease affects males and females equally, and it affects about one person out of every 12,000 to 20,000 people in a population. And in this picture here, you can see a couple of different patients with Angelman syndrome, which includes females and males. So epigenetics and lethal forms. So because imprinting is such a big player in this disease, because it makes the maternal gene so vulnerable to changes, we can definitely say that epigenetic modifiers, because they allow for genomic imprinting, we can definitely say that epigenetics play a big role in this disease, because with processes such as DNA methylation, histone modification, and RNA and other epigenetic processes, you have genomic imprinting in the first place. And here you can see in this picture a basic methylation process where a methyl group is added. So I found no website that directly stated that there were no lethal forms of this disease, but I came to this conclusion because only one copy of the UBE3A gene is activated, and therefore if there were lethal forms, then you wouldn't have patients of Angelman syndrome because they would have passed away. And also these patients have relatively normal lifespans, so therefore I concluded that there are no lethal forms. So then now on to Thomas, the prognosis of Thomas, my patient. These are some of the symptoms that she had: feeding problems, poor development, floppiness, seizures, unable to speak. She had a wide mouth, widely spaced teeth, a large protruding tongue. She frequently clapped her hands, locked her arms at the elbow when she was walking, so flexed arms. She drooled a lot, and she laughed inappropriately. So again, these symptoms very much overlap with other diseases, so we had to look a little bit further. And when we look at the karyotype, what we can see here is that all these chromosomes look relatively normal. But when we look at chromosome 15, even though the bottom part here looks okay, we can see that the top part of chromosome 15 looks abnormal. And that already gives us a hint that this is probably Angelman syndrome, but there's also other diseases, other genetic diseases such as Prader-Willi syndrome that also involve chromosome 15. So what really turned the light on, what really made me realize that it was Angelman syndrome with my research, was that the EEG analysis and the EEG test results show that there were abnormally high amplitudes and abnormally high spikes in the frontal lobe of the brain in Thomas. And that kind of led me to the conclusion with research that it was Angelman syndrome, and that's how I found out that the disease happens through imprinting. And that makes sense when we look at the methylation results, because when we look at these methylation results compared to a normal person, there's no levels of methylation for the maternal gene or the maternal copy of the UBE3A gene. And that basically tells us that the maternal gene is silenced. And then when you have paternal imprinting already going on and the maternal gene is silenced, then of course that's going to lead to Angelman syndrome. So that makes sense, kind of looking back with the knowledge that it is in Angelman syndrome. And then now the inheritance. And again, because the UBE3A gene of Thomas was silenced and not mutated, and because the inheritance of this disease is so complex, we really cannot make a conclusion about the inheritance with this pedigree here. But I did make a few hypotheses. For instance, this male here, he could be a carrier and he could pass on the mutated allele to his daughter, and then that's the mother of Thomas, and she could have passed it on to Thomas, making her, because that's the only activated gene of UBE3A from the mother that could have caused Thomas to become affected. And the same thing you kind of see here with Thomas's mother's aunt. She has two handicapped children. The same thing might have happened here with the carrier here, her father. But again, that's a little bit unclear. It's a complicated disease, so we can't accurately draw a conclusion here with this pedigree. But these are just some hypotheses. Thank you, guys.